We are on the ancestral homeland of the Ho-Chunk Nation, who call this place Dejope since time immemorial. The UW-Madison, our shared future marker on Bascom Hill, tells how this sovereign nation faced decades of forced removal and ethnic cleansing. We celebrate and recognize today's vibrant culture and the ongoing contributions of the Ho-Chunk Nation and all 12 of the First Nations here in Wisconsin. We're honored that Indigenous scholars, elders, and communities are willing to share knowledge that leads us all towards a more equitable and sustainable future. Now, as the landscape for health research and global health initiatives drastically changes in the U.S. and abroad, this year's symposium theme, Why Global Health Networks Matter, reminds us of our collective responsibility to keep moving forward by establishing relationships across disciplines, sectors, and geographies. In an interconnected world, global health networks are essential for advancing research, improving health outcomes, and creating stronger, more resilient health systems here in Wisconsin and abroad. I'm going to use the old man glasses here for a little while. This evening, we'll hear from a diverse panel of researchers, wildlife experts, and public health officials who are going to discuss why global health networks matter, using the example of white nose syndrome in bats. The decline of bat populations due to white nose syndrome is a really important issue for health in both humans and our environment because of the cascading effects on agriculture, food chains, biodiversity, and of course, public health. Protecting bats is not just about preserving a species, but also safeguarding environmental services that bats provide, which provide benefit for human health, food security, and our environment. As we're going to learn tonight by uniting efforts across disciplines to understand the One Health connections with human, animal, and environmental health, we're going to begin to understand why health networks are essential in combating white nose syndrome and mitigating its long-term ecological and human impact. Following the esteemed panel, and I'll introduce them and you'll get to know them very well in the near future, we're going to be honored to welcome Apurva Mandavili, a science and global health reporter for the New York Times, as our keynote speaker, and fortunately a former Badger. I guess once you're a Badger, you're always a Badger, who's going to discuss her experiences reporting on public and global health issues and collecting stories across health communication networks. As I mentioned, Apurva is a Badger alum, and we're really thrilled that she was able to join us for tonight. As a leading research institution, UW-Madison thrives by leveraging its vast networks to connect faculty, students, and resources with experts and organizations across the globe. Tonight's symposium would not be possible without the guidance and network of our co-hosts. That includes my team over at the Office of Global Health at SMPH, the teams at CALS Global and the Global Health Institute staff, the advisory committee, and the Board of Visitors. We also want to thank our generous sponsors and collaborators across the Center for South Asia and the Latin American, Caribbean, and Iberian Studies program, and everybody who submitted poster abstracts to share with us tonight. And of course, we also want to acknowledge and thank all of you, both here in person and online watching in the hybrid format, for joining us tonight to broaden our network of global health enthusiasts. A couple quick housekeeping things. Please silence your cell phones. Following the fireside chat and the keynote, there will be a time for Q&A. So during that time, we'll have some microphones circulating here in the room. But if you're watching online, please put those questions into the chat, and some smart person who's monitoring that will be keeping track of those and reading those for us. And the event is being recorded for your colleagues who are unable to join us. This will be available on the GHI website in the very near future. All right, so without further ado, unless there are other housekeeping things that I need to know about, nope, we're good. I get to introduce our panel and our moderator. First, I'll introduce my colleague and friend, Jorge Osorio, who's going to be serving as our moderator. As I think everybody in this room knows, Jorge is the Institute Director at the Global Health Institute. He's a professor in the Department of Pathobiological Sciences over in the veterinary school and has a lengthy career in both medical sciences, as well as virology, the field of epidemiologic studies, vaccinology, antivirals, and vector control programs. So as an infectious disease vaccine nerd, I'm a huge fan. He helped found the Columbia-Wisconsin One Health Consortium, which is a joint effort between the University of Wisconsin and the Universidad Nacional in Colombia. The center was renamed the GHI One Health Center, Colombia, and really serves as the gold standard for similar projects now that are being established in Mexico, India, Rwanda, and South Africa. Jorge also founded Vaxthera, which is a Colombian-based company that produces vaccines and biologicals for Colombia and for the region. He's also a co-founder and chief scientific officer in Envirogen, which is a biotech company that developed novel chimeric tetravalent dengue vaccines. He's also developed vaccines against chikungunya, influenza, rabies, plague, and many other infectious diseases. In his spare time, he sleeps. So join me in welcoming Jorge up to the stage. Next up, I have the pleasure of introducing my new friend, Eyal Frank, who's an assistant professor at the University of Chicago Harris School of Public Policy. Eyal is an environmental economist, and he works at the intersection of ecology and economics. His work addresses three broad questions. How do natural inputs, namely animals, contribute to different functions of interest? How do market dynamics reduce natural habitats and lead to declining wildlife population levels? And what are the costs, indirect in particular, of conservation policies? These areas of research present a causal inference challenge, as manipulating ecosystems and species at large scales is often infeasible. In his work, Dr. Frank draws natural experiments from ecology and policy and uses econometric techniques to estimate different pieces of the puzzle regarding the social cost of biodiversity losses. Dr. Frank's September 2024 published piece in Science, which I ran across and made my head explode a little bit, is really the foundation and basis in many ways for this panel, because while I knew about a lot of people working around this topic here on campus, his work really brought this all together in a really novel way and shared an example of how biodiversity loss affects human well-being, and he presented observational methods for quantifying these costs. So welcome, Dr. Frank, and please join us. Next, I've got the really distinct pleasure of introducing my friend and colleague, Dr. Bruce Klein. Bruce is a tenured professor and chief of the Division of Infectious Diseases in the Department of Pediatrics, and one of my many bosses on this campus, over at the University of Wisconsin School of Medicine and Public Health. Dr. Klein's got an abiding commitment to service. He's served as the division head in pediatric infectious disease for over 2 decades. He's had all sorts of leadership positions in the Department of Pediatrics, including vice chair for research for several years. He's really committed to faculty career development. There are countless lives in people's careers across this campus that he's impacted, and he's served on many of the tenure and promotion committees. He's chaired the university's Divisional Committee on Biological Sciences, the Med School's Committee on Promotion to Professorship. He's held leadership positions nationally at the American Society of Microbiology, Medical Mycology, the Gordon Research Conference on Immunology of Fungal Infections, and a charter member of multiple NIH study sections, including service as chair. He's a fascinating guy. He's also on the athletic board at UW, so if you get bored talking about either fungal disease or pediatrics, he's got all sorts of interesting stories about athletics at UW as well. In July 2024, Bruce published a piece in Science with Marcos Isidoro Aiza, a PhD candidate in his lab, and they shared their work uncovering for the first time how white nose syndrome in bats actually gains entry and initiates the infection. So what you're going to see tonight is really a number of experts connecting dots about something that is really a much bigger problem than I think any of us could imagine. So welcome, Bruce, up to joining us. Next up is Dr. Tony Rock. Dr. Rock's a research scientist at the U.S. Geological Survey's National Wildlife Health Center in Madison, Wisconsin. Dr. Rock has contributed knowledge and management tools for a variety of wildlife health issues in numerous species, including avian botulism and lead poisoning in wild waterfowl, sylvatic plague, which is Yersinia pestis in black-footed ferrets and prairie dogs, as well as rabies in vampire bats. Just that statement makes me pause. Rabies in vampire bats. And most recently, white nose syndrome, the fungal disease that's been decimating insectivorous bat populations throughout the US and Canada. She's developing and applying novel methods to mass vaccinate wild animals against disease and has led large, intensive, and multi-agency field efficacy studies, including bait-delivered sylvatic plague vaccine to prevent disease in prairie dogs and orotopical application of vaccine to bats to mitigate white nose syndrome and rabies. She's regularly consulted on vaccinations of endangered species, for example, these black-footed ferrets for plague and SARS-CoV-2, as well as highly pathogenic avian influenza and California condors. Dr. Rock has served as the president of the Wildlife Disease Association and a review editor for the journal EcoHealth. She has more than 40 years of experience as a research scientist, more than 130 scientific publications in international journals. Happy to welcome you to join the group. And last, but certainly not least, is Dr. David Drake. Dr. Drake is a professor and extension wildlife specialist in the Department of Forest and Wildlife Ecology here at the University of Wisconsin-Madison. As part of the Van Deelen Drake Lab, his applied ecological research and extension programs primarily focus on wildlife damage management to better understand, manage, and conserve animal populations in natural and urban landscapes. So let's give a warm welcome to David as he joins the group, and to all of our panelists as I hand things over to Jorge. So Jorge is going to moderate and lead this really interesting conversation about white nose syndrome in bats and One Health.
Thank you, Jim.
You got it.
Okay, can you hear me? Yes, good. Well, thank you everyone for coming, and thank you for the panelist members for joining us, and welcome to our 20th Global Health Symposium. We're very excited about the event, and what I'd like to have here is a very good conversation about why networks matter, and also the importance of wildlife and One Health. Thank you. So why don't we start by setting up the stage? One Health. You know, what is One Health? I think we all hear about the concept of One Health, but I think what we want to do here is to talk about the importance of having healthy ecosystems, healthy animals, and healthy individuals. And that's what the concept of One Health is. And the idea is why University of Wisconsin can do really well with the embracing One Health. So we have here a great panel of experts. And first, I'd like to thank Il for coming all the way from Chicago. You know, thank you so much for coming to Madison. This is your, you mentioned your second visit to Madison. Good, thank you so much.
My pleasure.
Great, and then a great panel of experts that we have. What I'd like to do is start by maybe each one of you tell us about the work that you do and why the work that you do matters, for example. And we'll start with Dave, for example. You work in wildlife, and if you can tell us about bats, you know, why bats matter. You know, I think Jim said that he now is more fan of bats, you know, thank you, Jim. And so why bats matter and what's bat importance in the world, if you can start from there.
Yeah, sure. Thank you very much. And thanks for the invitation for the panel. So I don't research bats relative to white nose syndrome like the rest of the panel up here, but I have in my previous career looked at bats and specifically bat impacts from wind energy. And that is a pretty strong consequence of mortality in bats, and especially bats here in Wisconsin suffer little mortality from predation, and so, primarily suffer mortality from things like wind turbines and a disease like white nose syndrome. And one of the things that is so significant about mortality events with bats is, as a profession, we really don't know much about bats. We know roughly about bat diversity around the world, but we don't know much about demographics of bats. We know they're long-lived. They have very slow reproduction, so they may have 1 to 3 pups every year or 2, depending on the species. But we don't know much about pup survival into adulthood. We don't know much about how long adult bats survive and things like that. And so these rapid decreases in bat populations that we're seeing, either as a result of wind energy or a disease like white nose syndrome, are relatively alarming, because we don't know if reproduction, for example, is able to counter a lot of that mortality and see what's happening with that. So I think this type of a panel is really valuable to start putting more comprehensive information together in order to understand more about bat population and sustainability. And although I don't do research right now relative to bats, in my extension role, I communicate a lot and have a lot of interaction with the general public, both in the state and outside the state. And there is growing fascination about bats. I remember when I was a kid, my grandmother said she was walking home from the hair salon and a bat got in her hair. And as a 5-year-old, I believe that. Now I know my grandmother was lying to me, because bats, for one thing, aren't out during the day generally, and they're not going to go after my grandmother's hair, even if they were out during the day. But that just illustrates that a lot of people don't particularly have a favorable opinion about bats. And I think that one of the things that white nose syndrome has done is it's provided a higher profile to bats, and I think it's changed people's perception of bats, which I think is a good thing.
Excellent. Thank you so much. So yeah, now that know that bats matter, we hear about white nose syndrome. And Tony, if you don't mind, tell us exactly what is white nose syndrome. You know what it consists of.
Sure.
Tell
And
us.
also, thanks for the invitation to be here today. So white nose syndrome is a disease that only afflicts bats. It's caused by a cold-loving fungus, so it attacks bats when they're hibernating. That's when they're the most vulnerable, really, in their life cycle. They put a bunch of fat away to store to get them through the winter period so they can emerge in the spring and start eating insects again. And this fungus infects them during hibernation. It eats up their energy stores, so they either die outright or they fly out early looking for food, and they freeze to death. They don't find food. Some of them actually fly out and have this really strong inflammatory response. It affects their wings terribly. That's where it mostly infects the bats. Not so much, we see white fungus on their nose, but it's really the damage that it's doing to their wings that's important. And it can cause this really strong inflammation, and that inflammation can actually make the skin on their wings just slough off. They can't survive that either. So it's really been a decimating disease to bats in North America. We first detected it in North America in 2007, but it was probably already here for a year or two. That was in New York, and it has spread across the country since. And we now have white nose syndrome occurring in Washington, and the fungus is already in California and Idaho. So in this short period of time, relatively short period of time, it has moved across the US, killing bats, millions of bats. So it's really a serious disease. And as we know, bats form a lot of ecosystem functions, which we're going to get into more today. So it's been pretty critical and pretty devastating disease for North American wildlife.
And how did it get to the U.S.? How did it come here, and do we have it anywhere else in the world?
So it was first named and learned about in the US when it started causing disease here. It was actually identified by biologists at the National Wildlife Health Center, or diagnosed by them. And for a while, we didn't know what was causing it, but it was definitively determined to be this fungus there. Since then, people started looking elsewhere in the world, and the fungus is actually found in Europe and in Asia. And from genetic work, it looks like it originated in Asia, made its way to Europe, and then it came over to North America just recently. Probably it's not, again, this is just a hypothesis, we don't know for sure, but probably on people's spelunkers' shoes. And it's a fungus, so it makes spores that are pretty resistant to environmental conditions, so it can come in on clothing and gear. That's how we suspect that it got here, but we really don't know for sure. But we don't see that kind of disease in Asia or in Europe in bats at all. Perhaps they already went through, especially in Europe, through sort of a period of time where it killed bats, and those that were going to survive did. But we didn't really know anything about this disease until it came into North America.
Okay. Wow, very interesting. So I remind the audience to please think about questions and write your questions, and or the ones online as well do the same thing, and you know, and back we'll collect the questions. So now, Bruce, from a physician to now someone that loves bats, you know, and how that change came, and you and your team now even studying more in detail how bat cells get infected by the fungus. And this is such a fascinating work. So how did you become such a fan of fungal diseases we know, but also bad diseases as well?
Sure. So this has been an interesting and enjoyable journey. Actually, I would say thrilling journey. So as a physician scientist who sees patients, but also works in a basic research laboratory, we've had a long-standing, many-decade passion for studying fungi. And we are interested in the immunology and pathogenesis of fungal infectious diseases. In the past, mostly that affect humans, children and adults. And we're very interested in finding general principles of how mammalian hosts defend against these sort of wily, poorly understood pathogens, and how they do their deed, how they get into the host, and how they invade and evade the immune system. And so in looking for general principles that apply across different fungi and even the microbial kingdom, I think it leaves us open to other investigators who see what we found and get in touch with us. And so that provides opportunities for collaboration. And one day we made an interesting discovery in another fungus and published it in a disseminated journal, a widely disseminated journal. And I got a call out of nowhere from Tony or Jorge or both saying, we're working on a fungal disease problem and we wonder if you can help. And, I was aware of white nose syndrome, and I do have a background in epidemiology and public health in a former lifetime. And so any opportunity for translating discovery into patients or in a manner that impacts and enhances and improves public health always catches my eye. And I thought that this was a unique opportunity to really work on a problem of considerable public health impact. And so I was all in. Soon after I started working with them, one of the young trainees at the National Wildlife Health Center got wind of this, contacted me and asked if I could be his PhD supervisor to study white nose syndrome. And I thought, oh my goodness, what have I gotten myself into? But it was the best decision I ever made, and I haven't looked back.
Excellent. It's amazing how that goes. You're still having fun, right?
Great fun.
And then we got to EL because now you made the connection. We have bats, we have a fungal disease, we have child mortality. Tell us the story. How did you actually, how did this happen? All of a sudden you were enlightened or something, and then all of a sudden you saw Eureka, I discovered this. How did you become to associate of bat mortality with infant mortality? Please.
So I think that this, again, thank you so much for the invitation and the opportunity to share the story with everyone today. And I think it makes perfect sense for this question to come now after we've kind of like established the three pillars that my work is built on. We know that farmers have to deal with a lot of crop pests, and they will value any kind of like free biological pest control that they get. We know that ecologists have told us that like bats ate a lot of insects, and if they were to decline for some strange reason, farmers will have to compensate. And we know from the field of epidemiology and public health that we've been worried about pesticide use for a long time, ever since 1962, ever kind of like since Rachel Carson wrote the book on DDT. And, this is an alarming amount of like chemical pollution that we're introducing to the environment. And there are all of these different theoretical predictions. And in my field, in environmental economics, when we think about the natural world declining, we often ask, well, what can we substitute with? So if we're losing bats and we're losing on the biological pest control that they provide, what will farmers might do? And the optimal thing through that lens is like, oh, they're just going to use this human-made substitute. They're going to use more insecticides. And then it's like, that lines up with what ecologists said would happen, but then raises the concern about what about what all the epidemiologists and public health people are concerned about, what's going to happen? How can we actually trace that causal chain? And for what I hope are like obvious reasons, it's not something that lends itself well to a randomized control trial. Even if we're willing to embark on like a thought experiment of like mad science and say like, let's go out and manipulate bat populations, like let's kill a lot of bats here, but let's not kill them there. And let's decide that this county doubles their use of pesticides and this county doesn't. And if we do this at like large enough numbers, we can learn what is the true contribution of bats, what is the true contribution of pesticides to human health. But there are like, again, obvious feasibility and ethical constraints that prevent us from doing that. And in my discipline, we say, well, okay, just because, we can't go out and conduct an experiment, is there like a second best solution that can get us close enough to that? And we often think along the lines of natural experiments. So when I first learned about bats dying because of this invasive fungus disease, I already knew that bats are really important in stabilizing ecosystems and their biological pest control. And I knew all about the concern that epidemiologists had. So when I read that, I was like, hold on a second. This is a very rare kind of natural experiment. It started out in a county outside of Albany in New York. And it's been spreading ever since. We get this like every year we're turning on the switch of like bat mortality in other counties. And like every year there's like a new epicenter because someone brought the spores of the fungus to a new cave. And it's like kind of like as close as we can probably get to just like randomly going out and manipulating bat populations. So let's say if this prediction that ecologists had, do farmers respond the way that we think that they did? And we have pretty good data on pesticide use. And I was able to demonstrate that like in the years after the arrival of the fungus, we start seeing farmers use more of those insecticides relative to the years before bats started dying, and also relative how pesticides are being used in unaffected counties where bat die-offs are not happening. So we kind of had this like treatment group and control group, places that were experiencing white nose syndrome and its detrimental impacts on bats, and places that weren't. And we see that like this, prediction by ecologists that like, oh, farmers aren't just going to sit there while they're losing all of this pest control, they're going to compensate, pans out. Then it's like, okay, this is also then another good approximation to how like maybe an epidemiologist would think about the ideal experiment, even if it's not feasible. We have now these places that on this like timeline of the disease expanding from county to county are seeing this elevated use of insecticides. What can we do to say about the connection to human health? And often in environmental economics, we go and look at infant health or infant mortality simply because we think that those are like very vulnerable elements of society. And we think that even at low concentrations, they might manifest very adverse responses. And we don't have to worry too much or try to think about how to control for these like convoluted, long lifestyle choices and health histories. And there's a long literature that uses the metric, the outcome of infant mortality to learn about how bad is environmental pollution. So I just, again, like followed the footsteps of giants that came before me and like, okay, if all of the concerns about elevated insecticide use are real, we should potentially see a signal in infant mortality. And I was able to show that like infant mortality rates were increasing relative to places in the counties where white nose syndrome was occurring, relative to places where it wasn't. And I just want to be clear about this. This doesn't necessarily mean that in the places that were experiencing bad die-offs, that infant mortality was skyrocketing. Infant mortality in the US is fortunately relatively low, relative to historic levels or relative to levels in low and middle income countries, but it's enough that even a small change ends up being meaningful in relative terms because the baseline amount is kind of low. What it also doesn't mean is that it's not necessarily that infant mortality increased in those counties relative to years before. It's just that the overall trend is like declining infant mortality trends in the US, which is a good thing. It's just that the places that experienced exposure to white nose syndrome and elevated use of insecticides diverged from that declining trend. So they might be still, on a declining trend, but they're not declining as fast as the other counties that are not seeing this substitution towards chemical insecticides. So it was the use of this, what we call these best practice methods in the causal inference toolbox in applied microeconomics, all of these like big fancy buzzwords for just like a lot of different assumptions on the tool that we are, I think, all familiar with of like linear regression and of like trying to say like, here are how the predictions should pan out in the data. This is what we see in the data, and here's how we can then rule out a lot of potentially other alternative explanations.
Wow, that's fascinating, isn't it? Incredible. So must have been quite an amazing moment when you come up with all this together and start putting it all together, and what you found there fascinating. So I think from the point of view of the wildlife scientists and scientists here, we have to thank you because without that, people wouldn't pay attention to us at all. So, right? So And even Jim wouldn't pay attention to us that much anymore. But thank you so much for that enlightening moment. So when we go back, Dave, to think about wildlife and how human activities like we're describing here have an impact in the spread of infectious diseases, the spread of pathogens and so on. The work that you do in terms of wildlife as well, can tell us how the human interactions have a significant impact with wildlife and the spread of diseases.
Yeah, for sure. So there's a paper that was published in 2008 in Nature, and even though it's nearly 20 years old, I think that a couple of statistics in the paper are still relevant. And one of those statistics is that 60% of the emerging infectious diseases in the world are zoonotic. So that means they can jump species lines and especially into human populations. And 72% of those emerging infectious diseases originate in wildlife. So the biggest issue about the promotion of zoonotic diseases is when wildlife and humans come into close proximity. So you have a direct and indirect contact that potentially promotes zoonotic diseases. So two of the biggest issues that are promoting zoonotic disease across the world is, number one, is urbanization. And so in the United States, for example, 85% of the population that lives in the United States is living in an urban landscape. 50% of the world is currently urbanized, but by 2050, about 70% of the world is going to be urbanized. And so even though the urbanization has a relatively small footprint across the landmass, that the impacts of urbanization are pretty severe. And what happens then is that either as we urbanize and we build out, we are encroaching into wildlife habitat, and or wildlife are moving into the cities with us. And so we've got all these this density of human population juxtaposed on top of abundant, overly abundant in some cases, wildlife populations that are in relatively small bounded areas. And so there's a lot of both direct and indirect interactions between humans and wildlife. And most of those interactions are generally benign. But if we have a zoonotic disease outbreak, it doesn't take long for those interactions to accelerate through those dense populations of humans and wildlife. And so urbanization is a big one. And when we think about the way that, at least in America, that cities are developed, we develop cities first and foremost with humans in mind in terms of where are we going to house everybody and how are we going to move them as quickly as we possibly can across that urban landscape so they can get to the store or get their kids to school or get to work or whatever the case might be. And then as a retrofit, we look at putting green spaces in these urban landscapes. And a lot of times at that point, these urban green spaces are fit into areas where land is either accessible, so it's available to purchase, and it's affordable. And a lot of times where we put green spaces in urban landscapes is not the same place that if we thought about where is it best to put green space, that's where we'd put green space in a post thought. So what typically happens is we've got green spaces that people and wildlife are both using, sometimes at the same time, sometimes not. But instead, if we were to build cities with the forethought of both what's best for humans and for wildlife, or for humans and nature, perhaps we would create some kind of a green buffer where we had some separation between wildlife and humans. And we don't necessarily have that. So we have this juxtaposition of humans and wildlife, and it really provides a great opportunity for zoonotic disease to spread across humans. So that's one issue. And then the other is habitat destruction, and that comes in relation to urbanization, but it doesn't necessarily have to be related to urbanization. But when you destroy habitat or modify habitat for wildlife in a way that is not positive for wildlife, it causes a couple of things. Either the wildlife that are still there, they're going to concentrate into a smaller area. So you've got more closer interactions of wildlife. So if you've got a sick individual, that disease might be able to spread quicker through that wildlife population. And or as you destroy or modify habitat, those wildlife, if they don't stay there, then they're forced to leave that area if they want to survive. And a lot of times that pushes them into areas where we have also concentrations of human beings. So those are the two biggest ones. And then we also have issues like pet markets and pet trade. And although that is not a terribly common issue that promotes a lot of zoonotic disease, the most likely start to COVID-19 was at a pet market in China. So it doesn't take more than one incidence in order for it to have pretty consequential effects on that. And then other issues like climate change is another issue, whereas climate starts to warm up, You might get range expansions for both pathogens, but you also might get range expansions for wildlife. And so wildlife are going to then start moving into areas where perhaps you have greater densities of human beings as well. In addition to, you may have greater length and livelihood of that pathogen of wildlife and things of that nature. So those are some of the issues that create more positive potential for zoonotic disease spread.
Thank you, Dave. That's fascinating. So now that we have wildlife, Tony, you are an expert in wildlife vaccines, and you actually are developing vaccines for white nose syndrome, which seems to me really hard to do. Tell us the work that you do, how you go out in the field, this work that you do in caves and some other places, and how you come up with this and the type of work that you do in terms of testing those vaccines. And how do you know that it's working, actually?
Sure. Well, first of all, this was a huge team effort involving your lab, Bruce's lab, and my lab. And we all worked together. We had this idea that we might be able to, people were trying anything to try and mitigate white nose syndrome in bats. Lots of ideas about, chemicals in caves, antifungals on bats. Nothing was really very promising or working. And we had thought about vaccinating bats against white nose syndrome, but we started out with a simpler vaccine, rabies in bats. That's another very useful vaccine, and we use that as sort of proof of concept that we could actually vaccinate bats. And then that's when we called you, Bruce, and said, okay, now how do we vaccinate against a fungal disease? And based on some of your early research, we incorporated an antigen that we thought might work. We looked in the literature, found another one we thought might work, and we developed 2 different vaccines. And we really got lucky. I have to say, I think that we were very lucky in finding a combination that seemed to work in bats. And we first did studies in captivity. We brought bats into my lab and vaccinated them, challenged them with white nose. We put them in basically a refrigerator for the winter, which mimics their conditions in a natural cave. And then we looked at their response to vaccination, and we had definite positive outcome, that bats that were vaccinated survived at a much higher rate, had much less disease. It's really hard to keep bats in captivity. They just don't like it, and they need to be euthermic. They need to actually be awake to vaccinate them. So we immediately asked for permission. We have to get permission to do this kind of vaccine work from the USDA, Center for Veterinary Biologics, to do more testing in the field. They granted that in 2019 was the first time we vaccinated bats in northern Wisconsin at these large mines, big sand mines where hundreds of thousands of bats used to live. Their population had been quite reduced by the time we started from white nose syndrome. And we put pit tags in them so we can monitor individual survival. We put readers at the entrances to the cave, so every time they fly by, it records a bat. And from that, we were able to demonstrate that there was higher survival, more vaccinated bats passing those readers than unvaccinated bats when it came time to emerge from the cave. So at that point, we felt like we had a product that might work, and we started vaccinating bats across the country at different places. Sort of the leading edge of the disease right now is in the western states, and that's where we think the impact might be greater. We can talk a little bit about that more if you want later, why that is. And so we are now vaccinating bats in maternity colonies. We catch them, we just drop a drop in their mouth and put pit tags and other things on them, and then we let them go. Ultimately, our plan is to incorporate the vaccine into a spray that we can just spray it on the bats. They lick it off themselves. So that's how they're vaccinated orally. Ultimately, we would like a hands-off approach. We've also considered applying it to their structures, so when they go back in, they get it all over themselves, in a sticky substance. They, again, lick it off right away. They don't like anything on them, so they groom it off immediately and even groom each other and pass it around that way. So that's where we're at right now. The Fish and Wildlife Service is already using the vaccine in northern long-eared bats, which is a bat that was fairly common even here in Wisconsin, and it's now on the endangered species list from white nose syndrome. You know, it's one of the main species that we may lose because of white nose syndrome.
Yeah, and you work very closely with the local DNR authorities that are part
Yes,
of
of
your team.
course. Yeah.
So this is all very coordinated effort as well.
Right. We work with state wildlife biologists, Fish and Wildlife Service, Bat Conservation International at some places, lots of NGOs, universities. So
Excellent. Excellent. Wow. So Bruce, now you're part of this team and working in fungal diseases. How many vaccines do we have actually in humans for a fungal disease? How difficult is to make a vaccine for this? Because they are very unique pathogens, aren't they?
Yes. So there are currently zero vaccines for fungal diseases in the human population. There are probably experimental vaccines that people are studying in humans and then perhaps in wildlife as well as in domestic pets. It's challenging to create these vaccines probably for several reasons. One reason is that the organisms are very complex. They have huge genomes, many, many different antigens, and so the sifting and winnowing, to use a phrase appropriate for Badgerland, is very, very challenging to find a needle in a haystack that is a very potent stimulus of the immune system. That's one complication or challenge. A second one is that fungi are eukaryotes. They're one of the kingdoms of life, very closely related to human eukaryotes. And so when one finds a strong protein or antigen stimulus of immunity, sometimes it's shared very closely with humans, and certainly you wouldn't want to stimulate immunity in an individual that could cross-react. A third reason is that is commercial. Fungal diseases aren't as common and widespread. They're often considered niche pathogens, and so getting commercial interest to really invest in this and buy into the concept of a vaccine where they may not see the payoff financially is a third major challenge. A fourth one is that the field is relatively smaller, for example, than people that work on bacterial problems or viral diseases. And so, so that you really need a critical mass, as we've demonstrated in collaboration, to really make this go. So there are a number of different challenges that make the reality of vaccines a difficult one. There's probably a fourth one. The experimental vaccines that work effectively are often live attenuated vaccines. So there's a live attenuated vaccine that we've created for a fungal disease that's endemic in Wisconsin called blastomycosis. We have protected probably tens and thousands of mice with this vaccine in the laboratory, and we also know that it's highly immunogenic and likely protective in domestic animals, particularly in pets, dogs, where blastomycosis is a big problem. It's hard to move a vaccine like that into people because of the complexity of the organism and the hurdles and barriers of using a live attenuated vaccine that's so complex in a human population. There's also another vaccine that's a live attenuated vaccine for closely related disease, coccidioidomycosis, which is a problem in the southwestern United States. And that is also very effective, looks like in pets. And there's some effort to move it into human populations, but that will face considerable barriers as well.
Yeah, it's quite challenging. So, Il, when you put all these ideas, this story together, what lessons have you learned after seeing, we have and you like bats, because I've seen some videos of you with some bats populations and so on, and And I see the impact that the work that you do, but also economics. When you talk about, I think when you put money at the end of the science and the question is when people somehow pay attention. So what lessons have you learned from putting all this story together?
Well, I think that the kind of like if we try to summarize what are kind of maybe three broad takeaways from the work is one that like there is pretty, I think, good compelling evidence that bats are really important and provide with material benefits to human well-being. All of these predictions that ecologists have made and environmental economists have made and epidemiologists have made all have support in actual real-world field conditions of using insecticides and crop pests. And like we talked about earlier in the panel, there are many other threats and concerns when it comes to bat populations. This sort of like natural experiment with white nose syndrome tells us that it doesn't really matter what is the cause of bat declines, but when we lose bats, we lose on biological pest control, and instead we have to compensate, and that compensation brings about its own set of problems. So whether it's the expansion of wind energy or habitat loss or wildlife diseases, there are good reasons to think about investing more in the conservation and preservation and restoration of bat populations. And the other, I think, higher level, broader thing that emerges from the work is that when we think about do ecosystem functioning and services and biodiversity declines, does that at all matter for us as society, for human well-being? And a lot of people will say like, I don't know, bat populations are declining, species are going extinct left and right, my phone is still charged, my Wi-Fi connection is pretty stable, I'm doing okay. And I think that this kind of work shows that like there is value to these natural enemies and stabilizing each other and keeping each other in check in an ecosystem. It doesn't mean that every single species is going to be that important and potentially detrimental to health. But again, the concerns that ecologists have been kind of like voicing for years, I think should be taken more seriously by social scientists in a similar way to how it took a while for social scientists to start hearing what the climate scientists were saying about climate change is happening, it's going to be bad for humanity, not just this like thing that, matters for the weather report. And we took a while until we started seeing those analysis being produced. And finally, this is like, from less of an ecosystem or bat-related angle, but we do have regulations on the use of pesticides. When you start diving into what those regulations are, they seem a bit unsatisfactory in the sense of we test each compound individually. Sometimes we test it on rats, sometimes we test it on rabbits, sometimes we test for like exposure on the skin, exposure through respiratory mechanisms. Sometimes we see what happens when they ingest it. And we kind of like have all of this kind of like mishmash of like, we have established that it's like safe enough for that method of exposure, for that, non-human, but still mammal species that we think is like a good enough approximation. Out in the field, we're mixing roughly 500 different active ingredients, and we don't really understand what that mixture is going to look like. And we don't ask companies to test what the mixture effects are because, to be honest, it's kind of like an impossible thing to ask. It's kind of like an insane lift. And so we are somewhat reliant on these type of studies that use data from the field, that use like in situ observational data to tell us like, oh, maybe there is a cause for concern and maybe the individual regulatory approval doesn't capture that even in real world application levels, the combination, the kind of like a broad exposure to this like broad set of chemicals is potentially harmful. And it's hard to really figure out what's the threshold, which potential compound is more important when we do a pretty weak job of monitoring the concentrations of different insecticides or pesticides out in the environment. And that's going to be costly, but I think that this kind of work also shows that like there's value in monitoring the chemical pollution load that we discharge into the environment.
Fascinating. So going back with that last comment you made, and the theme of our symposium, that network matters, we have here members of government, still have a job, right? So,
For now.
far. The Department of Interior, academia from University of Wisconsin, different schools, wildlife ecology, medical microbiology, and the hospital, and University of Chicago, economics and data-driven research. How this type of networks could be potentially explored in the future and need to be done to enhance your work, and what would be without them? So this is a question I'm throwing in general to the panel. And we go academia. If I want you from University of Chicago, you made all the way here to come here. Thank you so much for doing that. But how seeing what you are seeing here in University of Wisconsin, you have seen going to campus, have to walk quite a bit.
Yeah.
How do you see the value of this the networking?
I think that, we're all part of this like big, gigantic academic network, and there's this tendency to narrow in your discipline and in your kind of like narrow field within the discipline and kind of like not see what's happening outside of it. And I think it's really important that we all read what people that are working on similar problems in other disciplines and in other fields are doing. I would have never even kind of like figured out that this mortality of bats was happening had I not gone on the US Geological Survey website and kind of like, oh, well, what are you working on? What are like some pressing problems? And I would not have realized that the decline of bats is potentially meaningful for human well-being had I not read a lot of papers in ecology and epidemiology to think that like, okay, there's the dot on bats and insecticides, there's the dots on insecticides and health, and there's like in my training in like applied econometrics, there's like this natural experiment that I can now connect the different arcs together. And I think that I've also really benefited not just reading a lot of papers outside of my discipline, but also getting to present the work to audiences where there were ecologists and epidemiologists and public health specialists that were able to like, give me the approval of like, you did well with like understanding the science behind what you're doing. Because that's something that like economists, bless their hearts, they're not experts on bat biology or public health and toxicology. So I think that like having forums that make us, engage in each other's work is really important.
Excellent. Bruce, what's your take on that?
Here on this campus, we use a sort of a buzz phrase of team science. And I think that bringing different people together with different areas of expertise creates flashpoints. And I think really strengthens the work. It really knits together the most powerful approach to a problem. I mean, when I think about how I got involved in this, getting a call from Tony Roque and picking up on a small observation I made and published, I thought, well, this is interesting. The US Geological Survey, the National Wildlife Health Center, I heard of it, but I thought, wow, here's an opportunity. And then working with you as well, an expert in vaccines in your own right. And so I just thought that this is a really rare opportunity to bring strengths from multiple disciplines on a really important public health problem. And so that the sort of the whole is far stronger than the sum of the parts. And that's how I see work like this. It really sort of, to coin another phrase, it takes a village, to really build a program with impact. And it's hard to go it alone, especially with really complex problems like this.
Excellent. Thank you. Your view from the government.
Yeah, sure. I mean, we totally rely on academia for the basic research, the things that we need to really understand to develop the technology. But federal scientists have a role to play too. I think it would be hard for university to have marshaled the forces we did to get out and vaccinate bats. And we've vaccinated about 5,000 or 6,000 so far as an experiment. We're giving some a placebo too to see what's, to prove that it's working. So we can do that kind of thing in the federal government pretty easily. That I think is harder for scientists in academia to do, but we couldn't do, we just completely rely on university research for much of the technology and are grateful to have partners to do that with.
Excellent. And Dave, what's your take?
I would agree with everybody that's been up here so far. I think obviously we live in a pretty complicated world, and there's a lot of complexity that we're trying to unravel. I think more powerful computing systems are helping right now understand large data sets, which is very helpful. Funding models, I think, are also important, and especially at the federal level, I think there's more funding opportunities that are requiring interdisciplinary research, which I think is important to do as well. But I think it's also, and I'm putting my extension role on here, I think it's also important that we communicate that to the public, because if the public doesn't understand the importance of this, they're not going to support it. They're not going to advocate with their politicians that more funding needs to be done, promoted for things like this. And so I think it's important that you do have a team effort, interdisciplinary effort, but I think it's also important that you communicate that to the public and help the public understand why this is important.
Excellent, thank you. How are we doing on time? Okay, do we have more time for one more round, or do we? Okay, excellent, because obviously it's hard to ignore the current situation, right, that we are right now. And I'm wondering, asking you, all of you, what else do you think needs to be done in your field to take it to the next level, and how the current situation might impact that, if it does at all or not, or maybe it's an opportunity. We have to see that there's less interest in sort of global health, saying that broadly, and we're very focused politically in the US. So how that impacts your work?
Well,
Put you on the spot.
Yeah, I think that like broadly in economics, there's been perhaps too much of a focus on work in North America and in the US in particular. And there's like, at least when it comes to like energy economics, there's this like joke about, the US is bigger than just California, because there's like so many papers just written on that. And so I think that something that might be good that comes out of all of the like turmoil now is that there might be a bit more work looking outside of the US, because the work will go to where like the data is not being wiped out, and like there's still data collection efforts, and there's like interest with local governments to like pursue these kind of projects. So we might be in a period that we get to learn more answers on these questions, and just like in a more global setting. And I think that might also be beneficial to have a bit more of that human capital getting injected to like other partner universities and centers.
Excellent. Bruce?
Well, one can't help make political comments in a climate like this. Part of what got me involved in this research was fundamental bench research. And, there was a recent article in the New York Times that basic discoveries supported by federal dollars yield enormous economic benefit. For every dollar invested, 5 are returned. And so I see it as incredibly valuable to do basic fundamental research that can spawn discoveries that can go in any number of different directions. And I just think it's unfortunate and short-sighted, that at a time when life has become, and the world has become so complicated, that we're really pulling back on a major investment that this country has made for probably 80 years that has really stood us in tremendous benefit, and also benefited people worldwide. So I would just say that I think we need to continue to invest in basic research and to use that knowledge to spawn cross-disciplinary, interdisciplinary teamwork together.
Thank you. Tony?
Yeah, I'll avoid the politics, but I see some really amazing benefits from this work, is that we know how we could vaccinate bats should we need to in the future for other zoonotic diseases. We're learning quite a lot about that, and we'll have those tools in place if needed. I believe. And I just want to bring up some good news. Bats are starting to recover in the East. We're seeing adaptation to this disease in the East and also in the Midwest. Wisconsin's a great example. We lost 90% of little brown bats in the state from white nose syndrome. But in the really large mines and really big hibernacula where there was, like I said, 100,000 bats hibernating there, they dropped down to about 20 or 30,000, and they're starting to come back up because there was enough diversity there that they could adapt. All the small hibernacula, small species that without such big numbers like little brown bats are not doing as well. And we view the vaccine sort of as a stopgap measure until they also can adapt. So one thing that I am interested in, you asked about more research, would be where do you stop then? You know, that's the question we're asking. If we are going to use it to help bats get over the hump, what do we need to learn about how they're adapting to this disease so we can then pull back and say, okay, we need to let bats adapt on their own? Because that's what we really want, right? That bats live on their own and we aren't handling them. So I thought I'd just end on that little piece of good news.
Thank you. Dave?
I'm going to turn to the theme of the conference or the symposium first and talk about One Health. And One Health, you were talking about, Jorge, is the medical community, human medical community, the animal community working together with the environment. And One Health has been around for a while, but it just doesn't seem like it's ever gotten the traction that it deserves. And I think that's maybe one area that would be very beneficial, is if we could promote One Health and get some more traction behind that. But the other thing, I'm going to turn inward a little bit, is I'm an urban ecologist. The reason I work in the urban landscape is because, as I mentioned, that's where most of the people live. That's where most of the human-wildlife interactions occur. And from a wildlife management standpoint, the vast majority of the state wildlife agencies that are responsible for managing wildlife, free-range wildlife within each state, they do not have a presence in the urban landscape. And I think that is a real tragedy, in the sense that that's where the vast majority of the constituency for the wildlife agency sits. That's where most of the interactions between wildlife and human are occurring, as I mentioned. And that's where a lot of the management needs are, not only from promoting positive management, for promoting promoting habitat management to benefit wildlife in the urban landscape, but also to manage a lot of the wildlife damage issues, and to proactively manage issues to combat zoonotic disease outbreaks and things like that. So I think that more emphasis on urban wildlife management would be very beneficial.
Great, thank you. So now we're going to move to the Q&A section. I think we're taking questions from the audience and also from online. If we have any questions. Bruce, we have microphones. Just 1 second. We're going to get you the microphone. It's coming. I knew you were going to have a question. So.
Okay, so we've got an insectivore mammal, and we have a disease that impacts them. And if you look at all of the analyses over the last 40 years, we've lost 45% of our insect population. And in Germany, a study that just came out, they've seen a reduction of 70% in flying insects, which is what the bats eat. So has anybody done any studies dealing with nutritional fitness going into hibernation and the ability to tolerate white nose syndrome? I mean, they obviously don't have the same food supply that they used to have 40 years ago. And so whether this is from insecticides or whatever, it has to have, at least I think it has to have some type of a negative impact on the bat population that relies on them for the food. So I'm just curious what's been done in terms of nutrition in white-nosed bat syndrome.
Well, we do know that bats that are heavier tend to do better, certainly with white nose syndrome, even within a species. And like big brown bats are hardly susceptible. They're twice the size of little brown. They do really well, bigger bats. But even within little brown bats, the heavier bats tend to do better. And so there have been a few programs. I can tell you that there's the Fat Bat program you saw on that video you shared, that people are trying to attract insects with lights and other things to places where they're hibernating so they can fatten up more. It's hard to measure the impact of that. Honestly, we've talked about how to do it, and it's really hard without catching a bunch of bats and tagging them and weighing them and all that, and sort of defeating the purpose, I think, of that. So, but it's a really good point, Bruce. And we do know there is a relationship between weight and outcomes for white nose syndrome. So weights in early hibernation when they go into hibernation.
Thank you. More questions? Jim? John?
Thanks, John. I'm not sure I'm allowed to ask questions, but I'm going to anyway. Is there, we constantly in the zoonotic world in infectious diseases are worried about viral transmission and obviously at some level bacterial, but are there other models for this type of fungal pathogen devastating or at least impacting animal populations that we could learn from beyond this one?
Or the environment in general, like what other examples do we have of fungal diseases?
Well, there's a massive die-off of amphibians across the world as a consequence of a fungal disease, chytrid, which is getting into all kinds of amphibian populations. Salamanders are dying as well from spreading fungal disease. Snake fungal disease is another entity that's getting into the snake population. So, I suspect that climate will also influence the growth of fungi in our environment and will continue to lead to new emerging fungal threats. Those are just a few that come to the top of my mind.
There's also coffee and banana plants that get attacked by different fungal pathogens. There's the story how I think in 19th century France, vineyards their roots got attacked by this fungal infection. And there's a research paper that shows how that resulted in lower height of men in adulthood because they had all of these conscription records. So places that were hit earlier and harder, people from those areas were a few centimeters shorter, as a proxy for nutrition and income. So we've experienced quite a few of these negative shocks, and in some cases rebounded well, in some not so well.
More questions? Any more questions? Yeah.
Hi, thank you all so much for being on the panel today. I really learned a lot. My question kind of relates to the point that was said about the need to engage the public in these types of efforts. What kind of efforts, if any, have been made to just engage general people, inform this about them, and what role can a general public play in helping address white nose syndrome?
Would you like to take that?
I'll start. So yeah, I think we try to promote information about white nose syndrome as much as we possibly can in every opportunity that comes up. And so I think just general public education to alert people that this is a disease that's going on. And again, Jorge, I apologize, I misunderstood your first question to me. I was supposed to talk about ecosystem services, but bats provide a tremendous amount of ecosystem services. And so one way is to personalize to people, to make them or help them understand, as the bat population declines, here's how it may directly impact you. So for example, bats are one of the primary pollinators of agave. Yeah. So if you drink tequila, you may not be able to drink as much tequila if there's not as many bats on the landscape. So somehow to draw a personal connection to people, to help them understand why is it important that I care about this and that I know about this, and then what can I do? And I think that what can I do is a little bit more difficult, especially for this type of a disease, because it attacks bats as they're hibernating in these hibernaculum. And so the biggest thing is, if you're a spelunker or a cave explorer, that you practice best management practices and you completely clean yourself in between cave events. You disinfect yourself and make sure that you're not transferring it from cave to cave, because that has been demonstrated that is a pretty primary form of moving that disease from an infected cave to a non-infected cave. So in that regard, there's not a lot that the general public can do unless you're in a cave, unless you want to, fatten the bats up by leaving your lights on and things like that. But it's also, there is persecution of bats, and we still have people who, when they see a bat, they kind of treat it like a snake, and any good bat is a dead bat. And so if we can stop that kind of mentality and that kind of action as well, I think would be beneficial.
How about from the wildlife perspective from the USGS?
So there's, we have events, Bat Week every year, as you might guess, around Halloween. And they talk a lot about different kinds of things the public can do, especially with education and putting up bat houses, learning how to construct bat houses and preserve bat habitat in particular. That would be the most thing. And just being cognizant, this whole idea that animals transmit disease to us, we don't transmit it to them. That is wrong.
Yeah.
We can transmit disease to animals, and we need to be more aware of that, and try to avoid handling wildlife whenever you can. And if you do, make sure you're wearing PPE to avoid both ways. We wear PPE to keep from giving, still do, to keep from giving bats coronavirus. That's during the pandemic, we had to wear N95s always when handling bats. And it wasn't the other direction, it was to prevent the bats from getting it. So,
And even bat immunology, Bruce, you're becoming now a fan of
yeah.
bat immunology, right?
I am. So why should we be interested in the bat's immune system? Well, here's an interesting sort of thought problem. There are a lot of similarities between hibernation and the immune system and metabolism with what happens during space travel. And I think we all anticipate that we may be populating other planets around the world, around the stratosphere, maybe not in our lifetime, but probably in the future. So the better we understand the immunometabolism of hibernation, I think the better we can really understand how to prepare and harness the immune system for the future of space travel. So that's not something I would have thought of until recently, but there are companies that are particularly interested in this question and the future of space travel.
Excellent. Fascinating. Any more questions that we have
I'll
from there?
only add on
Yeah,
that
yeah.
you can do something beneficial, not just for bats, but for species and biodiversity at large by supporting preservation and conservation of habitats. And every once in a while, a local municipality or township or county or state will have a vote about can we increase this tax rate, or can we issue a bond to support buying and protecting and conserving and restoring some habitats. And they don't always pass. So if you do get to vote, voting in favor provides protection for bats and many other species. And it's important to remember that we are getting something in return by doing that.
Thank you. That's fantastic.
I might just add on to that really quick. So wildlife in the United States is a public trust resource, and it's not a monetized commodity. And that is good for wildlife, but it also hurts wildlife. And one of the species that tend to do really well are species that have an advocacy group behind them. So Whitetail Unlimited or Ducks Unlimited, populations that are healthy enough that you can harvest a certain percentage of that population each year, and then people will donate money to habitat conservation and management and things like that. But without wildlife being a monetized commodity, people don't really care about it, and that's part of the problem with wildlife, is that there's no way to make money off of it directly, and without that monetization, it's hard to get people motivated sometimes to care about wildlife and conserve wildlife.
Absolutely. Johnny?
Hi, thank you. I'm Johnny Ullman. I'm in Population Health Sciences in School of Medicine, Public Health. Dr. Roki, quick question about selective pressures, possibly. Are you seeing, rather than, you mentioned adaptation, that they're somewhat rebounding. Is it potentially a diversity thing, or is it a genetic selective pressure? Are they going to maybe become, like you had said, with the native populations in Europe, potentially protected?
Well, we would hope so at some point. I'm not sure there's been quite enough time for that. I think some of the adaptation is tolerance of animals. You know, bats are fairly long-lived. So some of these are animals that have been in those caves for a while. So there's some immunological, resistance developing. We've been looking at the transcriptomics of bats that were infected, in New York, or bat populations from New York, Wisconsin, and then really newly emerging sites like Montana, and then comparing that to our vaccinated bats. And it's really interesting to see that the vaccinated bats are very similar to bats now in New York that have experienced the disease since 2006. So we're trying to get at what the exact mechanism is, but hopefully it will ultimately be, a genetic resistance. We're never going to get rid of this fungus. It's here to stay. They're going to have to learn to live with it. We can't vaccinate every bat ever, anything like that. So we're hopeful that they'll adapt just as those in Europe have done. And we'll try to protect those species that can't for whatever reason.
Yeah, I imagine like the front end of it as it moves across the United States. Is it more pathogenic, or is it
No,
as
it's
equally-
not. There may be certain species. There's some species we're quite worried about in the West. It's even smaller than a little brown bat. The Yuma bats seem particularly susceptible. We're seeing some differences in species susceptibility, but the organism hasn't changed. It's the same.
I'll leave one more
Okay.
quick one. The thermal limits
Yeah,
of
wait.
funguses.
Thank you. Well, we probably will continue the discussion later if you like, at the break. But thank you so much to the panelists. I think please give them a round of applause for such a great job done. Thank you so much. Thank you. Yep. Yeah.
I want to thank David Gregg, Tony Roki, Bruce Klein, A.L. Frank, and of course, Jorge Osorio for a wonderful job with this panel. I've now gotten over my fear of bats. I'm working on spiders and snakes, ticks, so I don't think it'll ever happen. But join me again in thanking our panel for a wonderful discussion.
Thank you.
And for those of you who are wondering or looking for a great documentary about fungal pathogens, apparently Last of Us is coming out with another season in the near future. We're going to take a short break now just to switch things over. And we'll be back in about 5 minutes once we have everything set up with our keynote speaker coming up online. So don't go far, and we'll take about 5 minutes. Thank you.
Thank you.
Hi, Apurva. It's Katie. Go ahead and test. But you're
Hi.
on full blast.
You can hear me okay?
No, hold on. Okay, try again, Apurva.
Can you hear me now? No?
Okay, sounds good. So stand by, Apurva. We appreciate you so much.
Hey, for folks out in the hallway, we're going to get started here in a moment. If you can start making your way to your seats, please. All right, welcome back, everybody. We're going to get rolling here. We got the technology figured out. It's my great pleasure to introduce Jennifer Kushner, who's the director of Cal's Global. We'll wait until you're all here. Great. Thanks for coming back in, everybody. We're looking forward to this next part. So it's my great pleasure to introduce Jennifer Kushner, who's the director of Cal's Global here at the University of Wisconsin-Madison. Jennifer leads and supports international engagement in agriculture and life sciences and has really become a coordinating leader across campus. Prior to joining Cal's, she was a state specialist with the University of Wisconsin Extension from 2007 to 2018. For the past 30 years, she's led and evaluated U.S.-based and global initiatives related to agriculture, the environment, and health. We're pleased to welcome Cal's Global as a symposium partner and look forward to the discussion with Apurva Mandivili. So Jennifer?
All right. Well, thank you all for being here. It's a pleasure to join GHI and the Office of Global Health at SMPH to host this important and milestone health symposium. We'll now continue with our keynote presentation. Please join me in welcoming Apurva Mandivili, Science and Global Health reporter for the New York Times. Apurva's reporting focuses mainly on infectious diseases like COVID-19 and Mpox, scourges like HIV, malaria, and tuberculosis that remain big killers in many parts of the world. She explores pathogens that cause these diseases, the tools that try to stop their spread, and the public health agencies that try to manage them. Apurva grew up in southern India, but lived in the United States to attend college in the Midwest at age 17. She goes back to India once a year to report on many of the endless public health stories and speaks 4 Indian languages, which she notes is an advantage when interviewing non-experts. She also speaks some French, which she admits can be helpful when reporting in Africa, and some Japanese. Apurva is a Badger alumna with advanced degrees in science and journalism, both from the University of Wisconsin-Madison and New York University. She has been writing and editing articles about science and health for more than 20 years and has been featured in The Atlantic, Slate, The New Yorker, and in the anthology Best American Science and Nature Writing. Apurva was also the founding editor-in-chief of Spectrum, an award-winning news site on autism science, before joining the New York Times in May 2020. She proudly shared in the New York Times 2021 Pulitzer Prize for Public Service for coverage of the pandemic, and was also a member of the team that was a finalist for the 2021 Pulitzer Prize for National Reporting. She's the 2019 winner of the Victor Cohen Prize for Excellence in Medical Science Reporting and has won numerous other awards for her writing. We are incredibly honored to welcome Apurva Mandivili as our 20th anniversary keynote speaker. Thank you, Apurva, for being here and for all of the incredible work that you do to shed light on science and health stories that connect us all. The floor is now yours.
Thank you so much for that really warm welcome. And it's, of course, my honor to be giving this talk at my alma mater. I have so much fondness for Madison, and I've been back a few times. And I have a son who's actually a sophomore in high school.
Hold on one moment, if you could. We're having a little technical issue.
Okay.
Stand by. Thank you so much, Apurva.
No problem.
Please test your mic again, and we'll hope it's working.
Can you hear me now? Yes? No?
A little bit better. Try one more sentence for us, please, if you don't mind.
Okay. Is this better? Is it the volume or the lack of audio at all?
So sorry, Apurva. Hold on one more moment for us, please. Thank you.
Sure.
Thanks for holding, Apurva. If you could try one more time for us.
Sure.
Thank you so-
Can you hear me?
Fantastic. Thank
Okay.
you so much.
Great. So you want me to start?
Yes, the floor is now yours. Thank you so
Okay.
much.
Okay. Well, I guess you have to have some technical difficulties. Anyway, I was saying thank you very much for this honor. It's my pleasure to give this talk, especially to the University of Wisconsin, because it's my alma mater. And I was saying that I have a son who is a sophomore in high school, and I'm trying very hard to convince him that Madison is where he wants to go. So we'll see how that works out. But in the meantime, I wanted to give you a sort of quick background on what I've been doing the last few years and how I think my work has really shown me anyway that global health networks really matter. So I'm going to share my slides and hope that you can all see them. Okay, so I wanted to start with a bit of history, actually, which is this is from 2002, 2003, when we were looking at the first SARS. And I like to show this slide because here I am quoting Tony Fauci saying, "We will do in this country whatever is necessary to contain the spread of the epidemic." And when I look back on this now, to me, it seems sort of naive and optimistic and sweet. And of course, at that time, we really did think that things could be that simple. And we saw how different countries responded really differently to SARS. But I think we had this idea that things would go a certain way if it had reached the United States. One of the things that I feel like we learned really very strongly from COVID is to move faster. And here, for me, it was incredibly important that we were not just looking at what was happening in the United States, what people were saying in the United States, but to really go beyond. And that reporting that I just showed you on SARS really came in handy for me when I was reporting on COVID, because I still had those contacts in Hong Kong and in mainland China and Malaysia and all these places where people were still working on these kinds of viruses in Vietnam and Thailand and Cambodia. And I was talking to them. And what I was hearing from them was that it was very clear that the coronavirus was airborne. And in fact, China had been talking about that since the end of January. And yet in the United States, we were not really hearing about it. And so this was a story that I was putting together for weeks, really, in March of 2020. And then right as the story was coming out, the CDC finally acknowledged that the virus, was perhaps not quite airborne. They didn't use those words for much, much longer than that, but that maybe people should start wearing masks and that people were spreading it without symptoms. And this story that I published much later in March 2023, I was finally able to piece together what had happened behind the scenes. This is a story I reported talking to one particular whistleblower from the Epidemic Intelligence Service at the CDC, and a bunch of people in that cohort who were still at the CDC and were speaking to me, anonymously, but was able to basically show that all through February, in fact, people at the CDC did know from their own investigations that the virus seemed to be airborne indoors. But there were various reasons, political and practical, for example, the run on masks, that made them not really come out with that information, even though they were asking their own people to quarantine for 2 weeks if they had been exposed to the virus, even in the absence of symptoms, because they were worried about asymptomatic transmission and spread of the virus through the air indoors. This is another way, I think, in which things moved very quickly during the pandemic, and it was not always comfortable for people. In the United States, before the pandemic, I had this rule that I would never cover preprints because they hadn't been through the peer review process. But of course, at this particular point, when everything was shut down, these preprints really became a source for us for information from all over the world. We had scientists from China, from all over the world, really, from all over Europe, putting in everything they were learning in real time onto these preprint servers. And this became the source of information. And so I still followed certain rules. I, didn't cover anything that seemed like it didn't quite make biological sense. I would run things by some sources first to make sure that they thought that it sort of passed the smell test, if you will. And then if it seemed like the methodology was solid and the finding made logical sense, then I would go forward and really sort of cover it. And so this is an example of where I wrote about a coronavirus variant that was spreading in New York. There were 2 different teams, one at Caltech and one at Columbia, both very well-regarded teams that had both independently come to this conclusion about this variant. The city knew about it, but wasn't ready to talk about it. And so my reporting was based on one preprint that was already out and one that was about to come out the next day. And we published this story. The city wasn't happy, but neither were some people online. And so I just wanted to give you an example of this. This was garbage reporting. There was a lot of real discomfort with the idea of reporters writing about preprints before the scientific community had weighed in on Twitter or wherever they wanted to. But, not everyone was treated this way. This is just an example I wanted to give of my colleague Carl Zimmer, who had written about a similar variant that was circulating in Los Angeles. And Carl's reporting was based actually not even on preprints, but on data that he had seen, but wasn't actually online yet. And yet he didn't actually get the pushback. So the lesson I learned from this is the reason that I'm not really on Twitter anymore. And I assume a lot of you probably are not either. Things got very unpleasant there. Communicate uncertainty. Again, I think this is where there were forked paths for public health professionals from all over the world and for journalists in some ways. And here again, I really relied on my global health networks to report things that I think we were a bit slower to come to if we had been only thinking about one country or the evidence from one country. So this is the first story I wrote about the coronavirus, and it was at the very beginning of February 2020. Because as it happens, I was a freelancer in early 2020, writing about infectious disease for the New York Times, but I wasn't yet a staffer. That they sort of pulled me in a couple of months down the line from that. But at this point, I wrote this as a freelancer. But I wrote this piece because I remembered that it was very rare, even with SARS, to see kids getting really sick. And we had seen similar patterns in MERS. And in January, I saw a paper in the New England Journal of Medicine from China that listed a bunch of patients, and there was nobody under 15 in that list who had been severely sick. And so it was based on piecing together some historical information, some information that we can sort of logically surmise about biological, phenomena, like that adults who have never been exposed to something might react more strongly than children, and also some early data coming out. And so this was, a way to sort of hint at this, but you'll see all the caveats, the way that we communicated that uncertainty with maybe freedom that public health professionals don't always feel, by saying why it mostly spares children. And so far, very few young children seem to be falling ill, and why we think that this pattern was seen in outbreaks of SARS and MERS too. So this was a way to get the information to our readers, but while still couching it with some uncertainty. And this idea of the entire world battling a pandemic together at the same time became very complicated for a lot of reasons, including the very different interpretations. We saw this, for example, with the COVID vaccines, when people were recommending one thing here in the United States, and they were recommending, for example, only one dose of the vaccine for kids of a certain age in Europe. We also saw this when there was the recommendation for vaccines for pregnant women. And so this was a case where there was very limited data on the safety of the vaccine in pregnant women. And the CDC looked at that data and said women should consult with their doctors and think about getting the vaccine. And the WHO looked at the very same data and had very different phrasing that's made it seem, against their intentions, I believe, as if they were saying that there's not enough information about the safety of this vaccine and that they were recommending against it. It was not their intention, but that's how it came across. And so that was very confusing for people to have these two global powerhouses in public health seemingly saying different things. So we did this story, and the next day, the WHO changed its phrasing and made it very similar to what the CDC had, which is, we don't have any reason to believe that there will be risk, which is what they had intended to say to begin with. And so communication became very important in the internet era, in the Twitter era, when everybody all over the world was dealing with the same problem. This is just another example I wanted to give of how, when you have these diseases crop up all the way across the world, it becomes very easy to make certain assumptions about things. And this is a case where I happened to be in Uganda at the time that this anthrax outbreak happened, and it was being reported as this giant outbreak of anthrax, and this was happening and that was happening. Well, I happened to be nearby, and so we drove to the town where this was supposedly happening. I got the reports from the county office, and it turns out that they had only really tested a couple of cases and had positives only on a couple. Obviously, it's not going to be possible for reporters or anybody really to be doing this kind of vetting in, I just happened to get lucky. But it's just to say that simply because something is happening far away, we can't just assume that all the information coming out is accurate, or that we are not seeing it through certain cultural filters. Tell the truth. I mean, as a journalist, I cannot think of a bigger lesson, a bigger truth for myself and for my profession. This became incredibly important during the pandemic for very direct things, like when initially the Trump administration was really presenting misinformation as the truth. So this was a case where you may remember in August 2020, we were told that people who did not have symptoms didn't need to get tested, didn't need to isolate. And that was just clearly wrong for anybody who understood anything about this virus at that point. And also, as one of my sources told me, the language that the guidance was written was like a Pulp Fiction novel, whereas CDC people normally sound like Shakespeare. And so this, I was able to show through my reporting that actually it was written by Brett Giroir, who was the deputy deputy secretary of HHS at that time, but it was being passed off as CDC guidance. And I saw emails, which I wrote about in this piece, telling CDC scientists that they did not have the ability to edit that information, even though they were all railing against it and knew that it was wrong. So sometimes truth-telling took that very direct form, and sometimes it meant really digging deep to find out why there was reluctance on the part of the global community to talk about certain things. Again, here, going back to the airborne thing, if you remember, we did not talk about the virus being airborne, and the WHO did not really acknowledge it until July 2020. So this was a story I had been working on for several weeks before that. As you know, in the global health community, the WHO is sort of king. And people were really very, very nervous about saying anything against the WHO. So it took a long time to do this reporting, but I was able to get some internal emails, some recordings of internal meetings. And what became obvious is that they had actually been listening to experts from all over the world. They had invited experts into the WHO to talk about how the virus might be spreading indoors through the air to particle scientists like Lindsay Marr and Donald Milton and people like that. But they had not gotten past their hand hygiene bias because of really just a couple of people on their infection prevention control committee. And so it was really instructive to see how you could have this global community of experts, 239 of them, all arguing that the virus was airborne. And yet this one small committee with power at the WHO, and really chaired by a couple of people, one person in particular, but a couple of people with power, was able to continue to advocate just for hand hygiene. And one of the things I discovered during the reporting is that they didn't want to recommend both hand hygiene and masks because they thought it might be too confusing for people. And so they decided to just choose one, and they went with hand hygiene. So this piece came out July 4th, 2020. Which was a Saturday. And then on Monday, Reuters asked the WHO, well, what do you make of this New York Times article? They said, oh, no, we are already actually reviewing this. And they said, we've been thinking about this anyway, and we've been looking at the evidence. And then a couple of days after that, they said, yeah, actually, the coronavirus can be airborne indoors. And as you know, you all in the global health field, who knows better than you that the WHO has so much power in the rest of the world. What the WHO says can make all the difference for policies, and it can literally mean a matter of life and death with the kinds of recommendations that results in individual countries. Sometimes we had public health experts really saying things that were a lot stronger than the data warranted. And at times like that, it fell to the media, again, to correct the record. So this was a case where Dr. Rochelle Walensky, who was then the director of the CDC, went on TV and said that vaccinated people do not carry the virus. They don't get sick. They don't spread it. And it was just a very strong statement, and one that we didn't really know. And if anything, in talking to respiratory disease virologists, respiratory virus experts, it was actually clear that it's probably unlikely to be the case that people may actually get sick again, that just would be mildly sick. But it was an example of where it became important all the time to place every piece of information we heard in the global landscape of all the information we knew, and not just what we were hearing from an official in any particular country or at any particular time. This is just to say that the CDC has continued down this path. And of course, we'll talk a little bit about what's going on right now in the Trump administration, part 2. But this was during the Biden administration, and the CDC was not actually publishing a lot of the data that it collected. And it still hasn't published some of this data, including, to my big shock when I discovered it. They had data on who was in the hospital by vaccination status, age, sex, and race. And that would have been very useful information for the CDC advisors and the FDA advisors looking to make recommendations on who should get boosters. But they were worried that the data would fall into the wrong hands and be used for anti-vax propaganda, and they didn't want to acknowledge that vaccinated people were maybe ending up in the hospital sometimes, and so they chose not to say anything at all. And I would argue that hiding data like that actually leads to a sense among people that something is being kept from them, and it actually breeds mistrust, and it allows anti-vax propaganda to get in more effectively because people feel they might be lied to. So what is the lesson that I take from that directly? It's to trust the public. We heard over and over these very strong statements. This is not happening. That's not happening. Vaccinated people don't end up in the hospital. They don't get sick, things like that. But really, I think what the CDC acknowledged over time, and hopefully this is a lesson that we have learned through that pandemic, is that it's safer and more honest to say that the science is incomplete. This is a changing situation. This is all we know for now, and it may change. And I just wanted to show you a few headlines from articles I wrote during the pandemic to underscore this point of trust the public, because before the pandemic, I would have hesitated to think that people can handle these incredibly complicated topics, or that they're even interested in it. And yet I wrote about things like the cytokine storm. I really went deep geeked out into the science of it. This is an article about the CT value, the cycle threshold value of a PCR test, and what that tells you, and should we be relying on that? This was sort of when we first started talking about PCR tests versus rapid tests, and the really strong reaction that a lot of pathologists had against the use of rapid tests. But again, like incredibly technical topic. Why you shouldn't worry about maintaining coronavirus antibodies. And in this piece, I talked about B cells and T cells and the sort of real nuts and bolts of the immune system. And people loved this stuff. To me, that was sort of evidence that when they need that information, people want to know, and they should know. They have a right to know. This is a story actually that I wrote just a few months ago, and I wanted to show that we are still sort of continuing along this path of let's just not talk about the unpleasant things. This is, a piece that I did after talking to dozens of people who believe that they've been harmed by vaccines. That may not be true in any of their cases, may be true in some cases, may not be. I wasn't there to legislate that. That was really beyond the scope of the article. But the point was that I interviewed these people. I actually made it a point to interview only people with advanced science degrees. Some of them were, actually 5 of them were vaccine experts, whose names you probably know, although not all of them were willing to be on the record because of the sensitivity of this, but a couple of them were. Some of them, this person pictured here was actually a public health researcher, a nurse who had helped out with HIV vaccine trials. These are all really legitimate people, but they were being labeled as anti-vax simply for bringing up the possibility that their symptoms might be related to the COVID vaccine. And I wanted to show you the example of the different ways that people could react. The top quote is from Janet Woodcock, who was then the FDA commissioner, and I spoke to her after, when she had stepped down, but for this article. But I saw how she had responded to some patients who had been reaching out to the FDA. So she said above, there still is not a lot of certainty about what causes symptoms people are experiencing. And the rest of her email was actually extremely empathetic, but she's acknowledging the uncertainty. She is trusting that person to be able to handle the fact that there isn't certainty, versus thank him for his email and cut him off. I won't tell you who that came from, but it came from a CDC official who was also being approached about somebody who believed that they had developed tinnitus as a result of the vaccine, and they were just being told to cut this person off instead of engaging in any kind of conversation. Know your role. Just wanted to mention this because throughout the pandemic, I got a lot of really angry emails from public health people who thought that I was undermining vaccines by raising any questions about them. This one, it says both knockouts, Moderna versus Pfizer, but the article was talking about data that was starting to show that Moderna seemed to last longer and packed a bigger punch. which is important information to know for people who are immunocompromised, who are older, who may need that bigger punch to maintain a strong immune response. And yet apparently just raising this was enough that I got an email from one public health expert who said that I should be ashamed and how do I sleep at night, things like that. My role as a journalist is not to convince people to get vaccinated. My role is to give them all of the information that they need, and then they get to decide what they want to do. Obviously, I don't want to undermine vaccines, but I thought this was a pretty straightforward undertaking. More recently, I've been spending a lot of time doing the kinds of reporting that I think I can contribute to, which is bringing sort of really hardcore science, talking about, what we know, looking globally. When you are a global health reporter, you see measles elsewhere, you see polio elsewhere. You know that just because those things are not here does not mean that they are nowhere. And you know what kinds of things can happen when you have a virus like H5N1 that's been causing huge problems in places like Cambodia. Here, we don't seem to be taking it very seriously. Talking about how the foreign aid cuts are setting the stage for disease outbreaks by cutting off, Mpox programs, Ebola programs, screening for diseases at airports, labs that need to be secured with pathogens, all kinds of issues, trying to really shed a light on that. And then I wanted to sort of give a sense of what kinds of things I think that we are going to see over time. What slow impacts are just, we have just sort of scratched the surface, and these are long-term stories that I see myself having to really keep an eye on. One, of course, is you may have seen that the Trump administration cut off about $12.5 billion altogether in federal aid to states. Most of that was from the CDC, some of it from SAMHSA, but the funding was for COVID in theory. It was started during the COVID era, but it was expanded to include all kinds of other things. And so states were using it to modernize their data systems, their childhood immunization systems, their data, disease tracking. Some states like Alaska were using it to automate their patient reports so that their epidemiologists didn't have to fill out everything manually. There was a lot of things. There were a lot of things that were really essential in these underfunded, antiquated health systems that states had pre-COVID that were all being modernized, and now that money has just disappeared suddenly. Apparently, there's now an order to restore the funding, but we've seen how these things have worked out with other restraining orders that from the courts. This Trump administration has not always agreed to them, or has agreed but has not actually released the funding. So one thing that I will be keeping an eye on is very much this sort of thing. What happens down the line when these state systems are deprived of the funding that they need? Another thing that I'm sure all of you in your fields have noticed is the really big slashing of funding for prevention. This is a story I did about how different it is now to look at HIV prevention. Of course, PEPFAR is entirely in question at the moment. Things seem to be sort of running with treatment, but PrEP was really cut across the board. Prevention was cut. And I just wrote a story yesterday about how they have actually gotten rid of the very last experts who knew anything about maternal to child transmission of HIV at the CDC, who were managing PEPFAR grants on those the vertical transmission studies and work abroad. And all of those experts now at the CDC, at USAID, Department of State, are all gone. So it's unclear how that work will continue. And so that, I think, is something that we really have to pay attention to, because even if the rest of PEPFAR functions till September with treatment, not having prevention, as you know, is a very big deal. And that's going to have really big repercussions. And then a third area that I'm going to be keeping an eye on is we lost a federal lab at the CDC that was tracking gonorrhea resistance. We also lost a hepatitis lab, but these are labs that are reference labs. The CDC lab has the largest repository of gonorrhea samples anywhere in the world. And they have all of these samples that they had actually still were processing when they were suddenly just fired. And so STIs are already a huge problem here. We know that there is gonorrhea resistance elsewhere in the world. We've seen cases, we've seen a couple here in the US, but as we know, there is no such thing as a border really when it comes to these kinds of things. And so we have lost in the United States the ability to know when something like that might come through the borders into the country. And then finally, I wanted to leave you with what kinds of things you could be thinking about, and I'm happy to talk more about this in the Q&A, because I think people generally tend to have a lot of questions about how the sausage gets made, and I'm happy to talk more about that. But one thing I wanted to mention is CDC restores, some purge files after gender ideology ban and outcry. CDC suggests terms like race and health equity off limits, then backtracks. Fired CDC scientists are reinstated for now. What do these things have in common? Is that in each of these cases, the media covered something in a big way, and there was immediate reaction. So in the case of the CDC site, we saw these web pages disappearing. We wrote about it. We wrote about which databases were starting to disappear, which websites were starting to disappear. And we saw that a lot of them started to come back. No, not all of them are back, but there were things like STI guidelines that had disappeared that came back that were very important. There were things like contraception guidelines that OBGYNs use all over to, refer to make their diagnosis. And when that became a big stink, those came back. These things, these words about race and health equity, those were sort of terms that were used in people's performance evaluations. And when we wrote about that, they sort of backtracked and said, no, people can use those terms. These fired CDC scientists, you may know that the Epidemic Intelligence Service was actually all of those scientists at the CDC, the sort of so-called disease detectives, they were on the list to be fired. And then there was a big pushback, and some of it would even reach Twitter. And Elon Musk was, tweeting back saying, "Oh, no, nothing's happened to them. They're fine." And sure enough, they were fine. And then a whole bunch of other young fellows were fired from the public health assistance program. With these young fellows from the CDC who are placed in public health departments all over the country, they were fired. Then there were some lab fellows that were fired. They were all reinstated after the outcry. Of course, we're seeing destruction at a pace that we've never seen before, and I'm not remotely suggesting that if you just make enough noise, everything will come back. But I do think that it is important to speak up. I think in the reporting over the last few weeks, almost all of my reporting has involved talking to people who didn't want to be named because they were worried about retaliation, and of course, I completely understand that. But I've also seen the trends changing, with people speaking up, with people wanting to be named, with people making a stink, because I think they are starting to notice that sometimes what you see is that when there is a big pushback, they don't like that attention, and they do restore the programs. And even in the very giant list of USAID, which awards are kept and which were terminated, there isn't a lot of rhyme or reason when you look through that list of what was kept. Really, it looks like what was kept are things that people advocated for very strongly in one way or another. So that's my sort of message to you. And with that, I'm gonna stop, and I'm just gonna leave you with this slide that, sorry about the very giant snake that you can probably see, but this was a story I did not that long ago. I went to Kenya, and it's a story I had been wanting to do for a very long time about snake bite as a big global health problem. And of course, I was consumed with COVID for years, but then I was able to go and tell the story. And then now, again, I'm facing a time when I don't think I'm going to have time to tell these kinds of stories. So even though it's a picture of a snake, to me, it is a picture that inspires me to look forward to a time when I will be able to tell global health stories that are not just driven by crisis and indiscriminate cutting of aid and so on. Thank you.
Thank you, Apurva, for your inspiring talk and incredible leadership. I know many of us here in this room and joining online are proud to call you a fellow Badger. I know I am. Let's turn now to the audience for some Q&A. I'll kick us off because I had one that in the waiting. I'll give people a moment to put their thoughts together. So the theme of today's event is why global health networks matter. And I am a social scientist by training. So we like to say relationships are core, they take a lot of work. My question to you is, what can global health networks do to be as robust and resilient as possible today, particularly in light of at times opposing forces to those networks being robust and resilient?
I think one of the things that I've been really impressed by in the last few weeks, and I know, I've been asked before, like, what makes you feel optimistic right now? And one of the things I've seen in the USAID community, for example, a lot of these USAID fellows, and the officers who were laid off, I see them sharing their information. And it's not just the global health people that I talk to, but also the people who are doing democracy and governance work, who were doing social sciences, who were doing, all kinds of work through USAID and through their partners coming together, and they have organized in a beautiful way, and they all have different expertise. So, you can reach out to these communities, and if they have figured out ahead of time, these are the people who will talk to the media. These are the people who will work on the lawsuits. These are the people who will put together, materials to help the public understand what these cuts mean. I think you're talking about a community of people who are used to seeing the worst situations and then thinking about how to make those better. So, in a way, this couldn't happen to people who are more able, with better skills to pull themselves out of the situation. But what I see in that is the way that they are pooling their skill sets and pooling their resources. I'm heartened to see countries starting to talk about how they can step up and take care of their own, but also help each other. I've already seen examples of certain countries where, they're starting to pull money from a different program to say, okay, we're going to rescue this particular, let's say, PrEP program until we can figure out a better solution. But there's a lot of creativity in these networks, and I think the best thing that the community can do right now is to share that knowledge and think about a future without aid in which there's more self-reliance, but also reliance on these networks.
Thank you. All right, looks like we've got another question.
Hi, Apoorva. This is Jim Conway. Thanks for doing this. We really appreciate it. And more importantly, thank you for all you do to speak up on behalf of science. In the topic of networks, and I'm not sure how much you can talk about this in detail, but when we're talking about how the sausage is made, one of the things people in your position rely on is your networks of information sources on both the scientific side, which obviously we're all easily accessible and want to be given opportunities to try to correct things that are incorrect in misinformation. But how does it work for you with your network of communications with people within the government, whether it's a friendly regime or an unfriendly regime, as far as getting the information you need to understand what's really happening behind the scenes without outing people or putting people into difficult situations?
Yeah, this isn't work that really just started just now. All through COVID, I had to rely on government scientists who, for example, during the 2020, during the first Trump administration, when Trump administration was handling COVID, that story that I showed about the Trump administration writing guidance that looked like it came from CDC scientists, that whole story was put together with government scientists who shared internal emails with me and who were able to say, this is what came through, this is what we would have wanted to change it to, and here's what they told us, that we couldn't even change this. So a lot of it came from those kinds of sources, but I will say I worked very hard at cultivating those sources during COVID because, as you can imagine, government scientists and government employees are very wary about speaking up and not speaking through their sort of communication channels of the press office and the clearances and all of that. But they spoke up because they felt like they needed the information to get out there. What's different this time is that really since the start of all of this happening, in fact, actually soon after the election, I got a few messages from people saying, hey, I'm inside the CDC. I think things are going to be bad. I want to send you information. Or do you want it? And that has continued. We have a tip line that at the New York Times where people can send in tips and editors look through those and assign them to the reporters that make sense for that tip. I've gotten tons of tips from CDC folks, but I've also gotten people connecting me to their friends. I know somebody else who wants to talk to you, et cetera. And at this point, I would say there are dozens and dozens of government employees who are talking to me who want to share information because they feel so frustrated with what's going on. Sometimes they're finding out information from me because they, for example, with the RIFs, the layoffs that happened last week at the HHS, they didn't even really understand what was happening to them or when things were starting to unravel. But because I had sources in all different branches of CDC, I was putting together information and understanding what was happening sort of more globally, and then being able to pass that information back to people. I mean, it's been a real experience. But I would say that it's true both of CDC and of USAID that people have been really open to talking, as long as their information is kept private, of course, and that we don't. I would like to say I'm pretty good at keeping people's information safe, and so that's been the number one promise, is that they will not be outed in some way.
Thank you. We have time for another question or two.
Hi. In the middle of your presentation, you gave examples of public health officials and scientists censoring information, trying to shut down discussion because they thought they were doing the right thing, the public couldn't handle it. As you talk to them now years later, do you have any sense that they understand that hubris may be part of why we're in the situation we're in now, where the public has lost trust in us as scientists and in public health officials? Or are we going to learn from that at all about how we treat this in the future?
I have a sort of mixed answer to that. So during MPOX, I felt that we were starting to make some of the same mistakes, and then that things pivoted pretty quickly. One of the things I saw happen, for example, is, we had Dimitri Daskalakis, who is a gay man, who was put in charge of the MPOX response, and he sort of understood how to talk to that community in very direct ways. So, we had information from the CDC on how to have safe sex in very blunt terms, which was sort of, very refreshing to see. It was the kind of communication they needed to do. And then they were going to sort of, pride gatherings and meeting people where they were and really sharing that information. It took a little while to get there, though. And we were talking about MPOX as if it could affect anybody for quite a while before they were sort of acknowledging more openly that, no, actually, it's this particular community that's most at risk. However, in reporting on bird flu, I would say we seem to have taken some steps back. In press briefings with the CDC and the USDA last year, we haven't had really any. We've had a couple this year since the Trump administration has come back. But last year, when the Biden administration was in power, we had these interagency press briefings with the USDA and the CDC, and often it seemed like they were more interested in maintaining calm than they were in sharing information. And so they would say, it's very low risk right now. It's very low risk to anybody, which is true, literally speaking. But in talking to experts, they would say, yes, it is very low risk right now. But the thing about bird flu is that things could change on a dime. And that part was not being communicated very clearly. And then the other thing is, they would talk about all the things that they were doing, like, for example, testing, but nothing was mandatory. And part of that is the government's hands are tied. They're not allowed to go into farms and force people to get, tested, for example. But in these briefings, they would constantly say, well, we're asking people to get tested. We're not forcing anybody. Nothing's being forced. No one is being made to do anything they don't want to do, as if that is the most important thing in a public health crisis, is that nobody is being asked to do anything they don't want to do. And so that it seemed like there's been very much this sense of like, we are not going to overstep ever again. We are not going to make people do anything ever again that they don't want to do, whether that's vaccine mandates or mask mandates. And that may be the reality that we live in. But I wouldn't say that I came away from those press briefings feeling like I was being given all of the honest information and being trusted to handle it.
Thank you. All right. Looks like maybe we should move. Thank you, Apurva, very much for your engaging keynote presentation and time this evening. We wish you continued success in your journalism, and UW-Madison will always be ready to provide scientific expertise. Let's give Apurva another round of applause.
Thank you very much. It was my pleasure.
Now I'd like to invite Jorge to the podium to help us kick off UW's Day of Giving.
Okay, well, thank you, Jennifer, and thank you, Apura, for such a great talk. And I think now we're going to talk about the information I want to share with you is the Day of the Badger. I think we all know what the Day of the Badger is. Do we or not? Huh? Yes? So I think what we want here is to share with you that it's very important for us to raise funds for GHI, for the Global Health Institute. We are a very small group. I think if you can stand up the members of the GHI, 1, 2 over here, and 2 down there. There's 3. There you go. So we are a very small group, and I think we really, in this day today, we need to really rely on some of you to support us because global health funding is going down, unfortunately. And I think we need to continue with this great work that we are doing with the team that we are working on and with sponsoring global health. So if you can take your phone, you can go to that, where we have it in the screen, and if you can take a picture, scan that, and please support us for the Day of the Badger. I think it's starting this week. When is it over? Until 24 hours, so it goes today and tomorrow. And then, so we hope to count on you to support us so we can continue making Jim happy, I can continue making Kellen happy, and everyone's happy, okay? Thank you so much. And I think with that, I think I'd like to now turn on to Jim to close the event, and thanks everyone for having such a great evening. Thank you.
Great. Thanks, Jorge. You messed up my papers here. Where was I? There I am. So a couple of last things, housekeeping. My gosh, you made a mess of this. There we go. So thanks to Jorge, first of all, for having shepherded GHI through such struggling and challenging times, and we really appreciate all of his leadership. So before we close, I would like to finish by acknowledging the small but mighty teams that are behind the Global Health Institute, the Office of Global Health, and CALS Global. Without these folks, really none of this would be possible. In particular, I'm going to call out somebody who's not somebody who usually likes to be called into attention, who I'm looking for. There she is. So I think most people in this room know Betsy Tegland. Thank you, Dr. Olson, for pointing out her as she tries to hide back there behind Dr. Berman. So I think it's not without any coincidence that this is the 20th Global Health Symposium, and Betsy Tegland has been here for all 20 of these. And in fact, much of the success that we've had is thanks to Betsy and her efforts throughout these many, many years. She has unfortunately chosen to chase the R word of retirement, which while many people around this room are failures at retirement and continue to be around, I believe that she is going to be a little bit more dedicated to it than many of the rest of this group. She is really one of the most extraordinary, compassionate, humble, kind, competent people I've ever met. We have benefited from her wisdom, her guidance, her incredible organizational abilities. In recognition, we nominated her, and we're thrilled that the campus and the entire university agreed. And she is going to be receiving the UW Martha Casey Award for Dedication to Excellence from the Chancellor in a few weeks. It's nice to know that people on this campus really do recognize greatness in our midst. For those of you that know her, you know how great she is. For those of you that don't know her, you missed out. But you do have an opportunity to meet her tonight and to get to know her. We are going to miss her desperately, but she's been really both the leader and the glue that's held much of this together. And if Betsy and the rest of the teams from all of our groups could please stand one more time. Katie, Kaylin, everybody else from CALS Global, from the Office of Global Health. We really appreciate all of you. So I think we'll wrap up there. I want to thank everyone again for attending. Thanks to all of our speakers for really remarkable contributions. And I learned so much, which is really the benefit of these kinds of gatherings and these kind of networks. We've got great food. We've got great music. We've got great posters outside. We've got drinks. I would like everybody to hang around, please network, get to know each other, visit our poster presenters. A lot of really interesting work being done. I'm going to ask the speakers and everybody to just stick around for a little bit. We're going to do some pictures. But thank you again for everybody for coming. We look forward to seeing everybody next year for number 21, and have a great evening. Thank you very much.